Welcome to AP Biology. This video is on the semi-conservative model of DNA replication. The current model of replication that we accept today is the semi-conservative model, and this model was proved correct by an experiment performed by two scientists named Meselson and Stahl. We'll be looking at the Meselson and Stahl experiment in just a moment. Here this diagram shows how semi-conservative replication takes place. We begin with one parental molecule of DNA, and you can see that this DNA is double-stranded. It's made up of two parental strands of DNA. During DNA replication, these two parental strands are going to separate, and two new daughter strands are going to be created. These daughter strands are shown in light blue. During DNA replication, one parental molecule of DNA is going to create two new daughter molecules of DNA. And each of these daughter molecules is going to contain one old parental strand and one new daughter strand. So this method of replication is called semi-conservative. It's called semi-conservative because during replication, the two parental strands separate, and each new DNA molecule, each new daughter molecule, is going to contain one old strand of DNA, and it's going to contain one new strand of DNA. Here you see an overview of how the Meselson and Stahl experiment worked. Meselson and Stahl incubated bacteria in two different isotopes of nitrogen. One being a heavy isotope called nitrogen-15, and second being a lighter isotope called nitrogen-14. Meselson and Stahl began by incubating bacteria in the heavy isotope nitrogen-15, so that all the DNA that the bacteria contained is going to contain this heavy nitrogen-15. So here you see one molecule of DNA, and you can see that both parental strands contain heavy nitrogen-15, shown in blue. Meselson and Stahl placed this DNA in a centrifuge, and you can see that you get one heavy band that is down low on the tube because it contains all heavy nitrogen-15. Meselson and Stahl then took the bacteria and placed them into the lighter isotope nitrogen-14. This way, all new DNA that was made would be made using the lighter isotope nitrogen-14. You can see that during DNA replication, the two parental strands separate. One of the old parental strands becomes half of the new molecule of DNA, and the new strand of DNA that was created using nitrogen-14. So when Meselson and Stahl placed this DNA in the centrifuge, it created a medium hybrid band in the middle of the test tube. It was a medium hybrid band because it contained one strand of the heavy nitrogen-15 DNA and one strand of the lighter nitrogen-14 DNA. Meselson and Stahl then let the bacteria go through a second round of replication, also in the nitrogen-14. Here you can see that one parental strand still stays around, making that heavy nitrogen-15 strand. And the new strand that was created contained nitrogen-14, shown in red. This nitrogen-14 strand stayed around. And again, the new strand that was made was also made using nitrogen-14. When Meselson and Stahl put this DNA into the centrifuge, it now created two bands. One that's still the medium hybrid band containing nitrogen-15 and 14, and a second band which was much higher because it was lighter. It contained 14-14. It created a light band. It's these centrifuge results that proved the semi-conservative model of replication to be correct over the two other proposed models. The two other proposed models were the conservative model and the dispersive model, And you can see here that Messelson and Stahl's results proved both of these models to be incorrect. The only possible model that could be true was the semi-conservative model. This concludes this video.